I welcome everybody to a new global IMMUNOTALK. It's noon here in New Haven, and I'm here with the co-organizer, Dr. Elina Zuniga, and our speaker today, Dario Zamboni, who will be introduced by Elina. Thank you.
Hi, everyone. Welcome. So it's my real pleasure to introduce to you Dr. Dario Zamboni. So Dario is native from Brazil, originally from Brasília, the capital, and now works in Ribeirão Preto, state of São Paulo, Brazil. So Dario is head of the Innate Immunity and Microbial Pathogenesis Laboratory at Ribeirão Preto Medical School, University of São Paulo. In 1994, he obtained his bachelor degree in biology from the University of Brasília. Afterward, he performed a PhD where he trained in microbiology, immunology, and parasitology at the Federal University of São Paulo, Brazil. Afterward, in 2003, he obtained a highly competitive Pew Latin American Fellowship to perform postdoctoral training at Yale University School of Medicine under the supervision of Dr. Craig Roy. There, as a postdoc, he investigated the innate immune recognition of microbes by pathogen recognition receptors. After 3 years as a postdoc, Dario decided to go back to Brazil, where he was appointed professor of microbiology at the Department of Cell Biology at the University of São Paulo, and quickly moved up through the ranks and now is full professor at the same university. Dario has received many honors for his important contributions. He was elected affiliate member of the Brazilian Academy of Sciences and affiliate member of the World Academy of Science for the Advancements of Science in Developing Countries. Since 2020, so just last year, Dario has been director of the Brazilian Society for Immunology. And since 2019, he is chair of the Brazilian nominating committee of the Pew program in biomedical sciences. So the same program that actually selected Dario and allowed him to perform his postdoctoral training at Yale University. As I mentioned, Dario has made many significant contributions related to the interaction of intracellular microbial pathogens with host cells. The pathogens under investigations included the intracellular parasite Leishmania and Trypanosoma cruzi, which caused major health problems in Latin America, and among other intracellular pathogens. He has provided very critical and useful insights on how the immune system detects microbial pathogens, particularly via the inflammasome system, and how the immune system and the inflammasome operates to clear infections. On the other hand, Dario has also helped us understand how intracellular pathogens subvert the host immune system to replicate and cause disease. His work has been published in top-tier journals, including Nature Medicine, Nature Immunology, PLOS Pathogen, and many others. His research program is funded not only by national grants, but also international grants that includes the World Health Organization and the Pew Charitable Trust. So more recently, Dario has expanded his research program, as many investigators across the world, to study coronaviruses, particularly SARS-CoV-2. And indeed, the title of his talk today is Innate Immunity and Inflammasome Activation in Response to SARS-CoV-2 Impacts the Outcome of COVID-19 in Patients. And we will learn more about that today, and we're looking forward to that. So, Dario, we are truly delighted to have you as actually our first global immuno speaker residing in Brazil. and representing Brazil. So thank you so much for joining us.
Thank you very much, Elena, Carla, and all the other organizers. It's really a pleasure to be here. It was a fantastic opportunity to present our research, and really appreciate the invitation to be here with you guys.
Thank you, Dario. It's really fantastic to have you. Before we start with the talk, since you have been so tremendously successful performing research in Brazil, going back to your home country and performing research, and have also trained a large number of students that have graduated from your laboratory, we were hoping that you could share your thoughts on what are the advantages and disadvantages of performing research in Latin America.
Yeah, so that's a very important question for all of us. It's not easy to do science in Latin America. There is many challenges, as there are challenges in many places in the world. In Latin America, we may have a little more difficulties, such as importation of goods and reagents, mouse for immunologists, animal facilities. There is always some challenging steps that we have to bypass in order to do science in Latin America. But I was always very certain about my decision to return to Latin America and establish a research lab here. And that happens because I believe the science is very important to the development of the countries, the societies, to the social advances that all countries need. And particularly in Latin America, we need more to make the information available to the people, to treatments, to diseases. And so we need more and more science. And I think that's an important mission that we as a scientists face every day, is like dealing with the problems of the society and try to use science and everything we learn during the training, the postdoc, the PhDs, and try to generate more and more science that can help the advance of the society. And in this sense, I think Latin America it's a fantastic region. I'm sure you guys are very familiar to Latin America. You guys are from Argentina. I'm aware of this very amazing country. And of course, not only Brazil and Argentina, but all the countries, for people that have been traveling through Latin America, and many, Peru, Chile, Bolivia, Mexico. That is an amazing country, amazing people. I'm passionate about the people in Latin America in general, not only Brazil and Argentina, but also in the other countries. And I think it's a joy to be like part of like a community that can help development of science and development of the society, of the communities. So I think it's worth establishing a lab here and try to give the chances of these amazing students that just ask us to join our labs as a initiation in science, and then they start for master. So you give the chances of these students to be scientists. I think it's an amazing thing. And different than soccer, which we love as a Brazilian, as Argentinians, if you have like a ball, Pelé and Maradona were born in every place of the world, they're gonna be a super amazing players of soccer, of course. But for science, you need to have a lab, you need to have a mentor. So it's important to have different labs spread through different regions. And I think it's one of our missions is like select these amazing students and give them the chance of becoming like a fantastic scientist. And of course, I'm very happy to foster this new generation of scientists in Latin America.
Yeah. That's fantastic, Dario. Thank you so much for doing that. So important to improve the science in the region, as you said, and for training many young scientists there. Thank you so much. And you get to live in Brazil, which I'm very jealous of. Anyway, so if you'd like to share your screen.
Okay.
So we can enjoy your talk.
Can you see my screen?
Perfect, perfect, yes.
So it's a pleasure to present our last data about inflammasome activation in response to SARS-CoV-2 infection in patients of COVID-19. So I will start with an introduction about the main question that my lab is focusing on, which is pretty much the key question that our immune system faces every day is like how to discriminate and recognize the microbes that can be pathogenic for us versus the non-pathogenic ones. We are surrounded by microbes. Our microbiota is amazing, the diversity, and most of these microbes doesn't do any harm to our body, but some of them can quickly invade our cells and gives us disease, and then you're going to have facing infectious diseases. And our immune system is very well prepared to sense these microbes. And many of you guys are going to say that the easy answer to this question is like, well, we have toll-like receptors, we have pattern recognition receptors, we can sense microbes and pathogens. But if you think about what is a component of a microbe versus a pathogenic microbe versus a non-pathogenic microbe, the toll-like receptors cannot really discriminate efficiently what is a pathogenic versus a non-pathogenic microbes. For instance, a pathogenic E. coli is very similar to a commensal E. coli in our intestines. So the toll-like receptors may not be the best receptors to discriminate what is a pathogenic versus a non-pathogenic microbes. And of course, we have, I'm sure you guys are aware of this information, we have a series of intracellular receptors that are ready to sense microbes as soon as they start invading our cells. They're going to somehow manipulate the cells of our body, the membranes of the vacuole, or secrete molecules inside the cytoplasm of our cells. And now we're going to give the chances of intracellular receptors to be activated. And these guys are the ones that can tell to our immune system that we are dealing with a pathogenic infection, and we need to mount an inflammatory response, a robust response that's going to recruit additional cells, and then it's going to produce the inflammation required for the adaptive immune to take place. And then I'm sure the intracellular receptors play a very critical role in recognition of specific recognition of pathogenic microbes, as opposed to microbes that does not cause any damage to our cells. And who are the cells? There is a series, huge families of intracellular receptors, and my lab is pretty focused on the investigation of the inflammasomes. There are many inflammasomes. Just to make the pictures more simple, I'm depicting three different inflammasomes here. One of them is the M2 inflammasome, then LRC4 nape inflammasome, and then LRP3 inflammasome. In common, they all forms aggregates in the cytoplasm of the cells once they are activated. The ligands are very different. So, for instance, M2 senses double-stranded DNA. The NAPE and LRC4 senses flagellin or components of the type 3 secretion system of gram-negative bacteria. And the NLRP3 inflammasome senses a damage in general in the cells, and when the potassium level goes down, there's signals to this inflammasome to become activated. They have adapter molecules, and they also have this common feature, which is activation of an inflammatory caspases. Once these platforms are activated, we have caspase 1 being activated in response to this different stimuli by these different inflammasomes. There are many more inflammasomes, and NLRP1 is a beautiful inflammasome as well. There is beautiful information, recent information about these receptors now, and many others inflammasomes, the pyrine, the NLRP12, NLRP6. There is many different platforms of the inflammasome that could be used in this introduction. I'm just to make the picture more simple, I'm depicting these three different inflammasomes to make the point that they sense some damage in the cells, and they activate a structure which we call puncta, which I'm going to explain later more in detail in the next slides, and they lead to caspase 1 activation. And of course, the NLRP3 inflammasome have gained much attention in the last years, because as this receptor senses damage in our cells, they are involved with many different inflammatory diseases. not only infectious disease such as bacteria, virus, fungi, parasites, but also gout, ischemia and perfusion, autoinflammatory diseases, diabetes, atherosclerosis, Alzheimer's disease, obesity. So we have a lot of situations in that our body senses damage. And as soon as there is damage in our body, membranes senses this damage. There is rupture of or opening channels in the membranes, and then the inflammasome, the NLRP3 inflammasome, starts to operate and produce inflammatory response in response to this during this different disease. So there's beautiful work, recent work by Mao and Hao that illustrated the bicreel IM, the structure of the NLRP3 inflammasome, in this case activated with the help of NEC7. So you see that once the receptor is activated, this is a single NLRP3 protein, and as soon as it's activated, there's the formation of this disc or ring-like structure in that the LLR domain of the receptor gets to the outside of the ring, and from in the inside part of the ring there is all the purine domain of this receptor. And then if we flip 90 degrees, there's going to be a disc or a coin-like shape of this receptor, and this is a beautiful view of the inflammasome activated NLRP3 inflammasome. Looking from the top of the platform, you see all this purple region is the purine domain, which is the terminal region of the receptor that is going to be important for binding to the ASC, which is a small molecule that is going to polymerize, and then is going to promote more sites for cleavage of caspase-1, and then the inflammasome is going to fulfill its function of activating caspase-1 and inflammatory caspases during activation of these platforms. During the infection, whatever happens is that the receptor is in an active form in our cells, in the cytoplasm of our cells, and as soon as there is a pathogenic microbe, and remember that it must be some pathogen that like messes up with the membranes or secrete molecules or causes some sort of damage in the cells, and then there's going to be signs to activation of this receptor, and now we have oligomerization of the receptor. There's this 11-fold NLRP3 disc is going to be formed, and as I mentioned before, there is the exposure of this purine domain of the NLRP3, and now purine domain is going to bind to the purine domain of ASC, which is the small purine CAR domain protein that is going to polymerize and make these long filaments of purine-purine interaction, forming these long ASC filaments, which is going to expose the CAR domains of this protein to the extracellular part of the external part of the filaments, and CAR domains are very effective to bind to other CAR domains, and now there is going to be the formation of these long structures which the CAR domains can bind to the other CAR domains, and then this can make this huge puncta-like structure, or speck. Some people say speck, some people say puncta, and there is this formation of this huge ASC speck or ASC puncta, which is going to provide multiple sites for cleavage of caspase-1, because the CAR domain are exposed to the extracellular part of the filaments, the external part of the filaments, and now procaspase-1, which is produced as a proenzyme, not active, it's activated in the filaments of the inflammasomes, and then now we have active caspase-1, which can start cleaving other substrates to promote inflammation. In the case of caspase-1 is going to very effectively cleave IL-1 pro, IL-1 beta, and pro IL-18, which are again produced as inactive cytokines. And as soon as they are cleaved by caspase-1, they're going to produce the active caspase-1 and the active IL-18. And now caspase-1 also cleaves a very important molecule for cell death, which is gasdermin. The gasdermins are a huge family of proteins. One of them is the most studied, gasdermin D, is cleaved by caspase-1, and the N-terminal region of the gasdermin D is going to punch pores in the membranes of the cells, and through these pores, the cleaved cytokines can be released, and this pore can allow influx of water, and there's this balance of the ion homeostasis of the cells swell and then lyses, and then this is going to release a lot of intracellular components in the outside of the cells, and this is going to be very important for induction of an inflammatory process. This may be very critical for recruit additional phagocytes to the site of infections to promote immunity against these infectious microbes that triggers activation of the inflammasomes. So this is trying to make a very simple process, which is activation of the inflammasome and induction of cell death and release of inflammatory cytokines such as IL-1 and IL-18. And this involves the formation of this speck, which is this aggregate of proteins in the cells. As an example of the ASC specs, I can show a picture made in the lab a few years ago. This was a research we are doing with a protozoan parasite, very important in Brazil, which is Leishmania. Leishmania causes a lot of disease, especially in poor people and in the less privileged regions in Brazil. And Leishmania is an intracellular protozoan parasite depicted in red here that lives inside the macrophages. And in this picture, this is a bone marrow derived macrophages expressing ASC GFP, fusion with GFP, so you can see the ASC dispersed through the cells in these non-infected cells. And as soon as Leishmania infected these cultures, we see two Leishmania parasites standing in red here. There is oligomerization, activation of the NLRP3 inflammasome, oligomerization of this ASC in a single speck in the cell. And now this inflammasome is going to be very important to restrict the replication of these parasites in the cells and to induce an inflammatory response, which may help the restriction of the parasite replication and return to the homeostasis. So the inflammasome may help in a lot of diseases. It's protective to a huge variety of bacteria, parasites, and virus, fungi. But also the inflammasome, once superactivated, they can cause damage, such as happens in inflammatory diseases. The superactivation, uncontrolled activation of the inflammasomes may lead to inflammatory situation in that they can cause some damage to the hosts. And in this line of investigation, about 2 years ago, I started moving a little bit the direction of my lab, not only to bacteria and parasites, but also to study virus, the response to inflammasomes to viral infections. And we had studied this important emerging virus, which is Mayaro virus. It's a very important virus, circulates in the Amazon region. And as soon as people is destroying the forest, and there is the habitat loss for many animals, there is an infection of humans with this Amazon specific virus, which is the Mayaro virus. And we have shown in this study that the NLRP3 inflammasome is very effectively activated in response to Mayaro virus infection. And also in patients with Mayaro fever, we see a lot of activated inflammasomes in these patients. And also I collaborated with a good colleague, which is Elden Nakaya, which is a bioinformat that did some system analysis. And then in chikungunya virus, it is another virus that causes arthritis. Is a very bad disease whenever it happens. And NLRP3 has also a very important signature in these patients and also plays an important role during this disease. So I would just want to make the point here that the inflammasome helps. It's a part of the innate immune response to many infectious diseases. But in some cases, it can worsen the infection because it can promote inflammatory disease, and this can be detrimental to the host and can lead to a more severe disease. So just to give an overview of how the inflammasome, the basics of the inflammasome actions and the role in protection and induction of a more inflammatory situation. And of course, about 1 year ago, we were all surprised by the fast spreading SARS-CoV-2, which causes COVID-19. And I'm sure this virus requires no introduction for this audience. But just yesterday, I checked how the status of the pandemic in the world, and the numbers are really bad. As you can see here, Brazil has more than 200,000 deaths. in the country, which is very severe, highly affected Brazil. There are many features by why that happens, but the response in Brazil are not appropriate as one would expect, especially there was a lot of negationist movements about the virus. And if you think Brazilian population is much smaller than US population, the situation in Brazil is actually worse than in the United States. And we're still facing a huge number of deaths every day in Brazil. And then at that time, in the beginning of the pandemic, I thought that as a mission, we should like stop our regular research and move towards the investigation of SARS-CoV-2 infection. And in Brazil, I'm hosted in a medical school. We had a lot of patients at that time. We still have a lot of patients now, but we had the lab running all the expertise in inflammasome, so why not focus the effort in investigating the role of the inflammasome in COVID-19 and in response to SARS-CoV-2. And then there was, of course, previous information about SARS-CoV-1, which triggers the inflammasome, the NLRP3 inflammasome. So this is a paper published before the COVID-19 pandemic. This was published with SARS-CoV-1, which shows that ORF3A is one of the non-structural protein of the virus. They activate an NLRP3 inflammasome. This was a paper published in January 2019, so before the SARS-CoV-2 emergence. And also there was this other paper also published before the pandemic showing that the ORF8B causes stress in the cells and activates the NLRP3 inflammasome. This is more complicated to do the translational because SARS-CoV-2 does not express a ORF8B. It expresses just one ORF8, which is right here. This is the genome of SARS-CoV-2. So as you can see here, the ORF8 is not cleaved in SARS-CoV-2 as it is cleaved in SARS-CoV-1. But that was the information. There was a couple of more papers showing that the coronavirus activates an NLRP3 inflammasome. And together with information of the first cases in Wuhan, we had this idea that the nature of the inflammatory disease supported the participation of the inflammasome during the COVID-19. This was an important paper that gave us the hint that the inflammasome were participating in the activation of the inflammasome during the COVID-19. There was a retrospective study with 21 patients from Wuhan, the early city that was affected by COVID-19. And so they identify a macrophage activation syndrome and cytokine storm as an important and associated with a disease severity. So there was some hints that the inflammation and the macrophage activation syndrome was associated with the disease severity. And there was also this other paper published in June 2020 with 107 patients also from Wuhan in the first cases of COVID-19, that they show that LDH, which is lactate dehydrogenase, it's a cytosolic enzyme which is released out of the cells when there is lysis of the cells. And they show in the study that the LDH was associated with disease severity. So it was linking cell death with LDH release, and LDH release is a marker of pyroptosis, which is a common feature of the inflammasome activation in the myeloid cells in general. That there is a lysis of the final stages is the gazamine D cleavage, and then lysis, the cell collapses and releases LDH release. So we had all the hints that like the inflammasome could be participating in the inflammasome activation in the SARS-CoV-2 infection and could be worsening the clinical features of the COVID-19 in patients. So I think by that time, in the beginning of the pandemic, there was a lot of labs closed. All the scientists started studying inflammasomes and or virus SARS-CoV-2, and there was a lot at that time. There was a lot of reviews about inflammasome and COVID-19. This was a PubMed search that I did a few months ago. You put inflammasome, SARS-CoV-2 or COVID-19, there's lots of reviews. So I felt that people start studying virus and what's happening. Oh, I had this idea, then maybe the inflammasome is activated. So as we did, many other scientists start looking for inflammasome activation in response to SARS-CoV-2 infection, just a lot of hypotheses, reviews, and so on. So we decided to start studying the in vitro infection with the SARS-CoV-2 to see if the inflammasome is in fact activated in response to the virus. And of course, that happens in the beginning of the pandemic. All the labs are shut down, and we had to use the P3 facility of the virology in our institution. And we got access to the first isolate of the Brazilian infection. This is one of the SARS-CoV-2 first isolates in Brazilian patients. And we established the infections in viral cells, and then we managed to infect primary human monocytes from donors, from volunteers, and we infected these monocytes with SARS-CoV-2. And what we did first was measure the punta formation. Remember, the inflammasome is activated, and there is aggregation of the NLRP3 and formation of the ASC spec together with NLRP3 spec. So we see this punta, which means inflammasome activation in response to infection. And you can see here, these are primary human monocytes infected with SARS-CoV-2 in vitro. You see NLRP3 stained in green. You see in some cells, you see a nice inflammasome activation in this cell. This is just a higher magnification of this region of this image. So you can score the percentage of the NLRP3 punta in response to viral infections. This is UV irradiated virus, so this death virus does not trigger inflammasome activation as expected. So we need, in order to get inflammasome activation, we need like a pathogenic microbe in general, and then as SARS-CoV-2 live virus is able to trigger punta NLRP3 punta formation, which means inflammasome activation. And in this experiment, we use it primate and unprimed cells. So the inflammasome activation, usually if you prime the cells, you upregulate all the receptors and the genes required for activation of the inflammasome. And so usually the inflammasome activation is much more efficient if you prime the cells. But for punta formation, even unprimed monocytes are able to trigger inflammasome activation in response to SARS-CoV-2. And in this bar right here, we inhibited NLRP3 with MCC 950, which is a small compound which is highly active and effective to inhibit NLRP3 activation. And you can see here, MCC 950 effectively inhibited inflammasome activation in response to SARS-CoV-2, suggesting that, supporting the notion that in fact this was really the inflammasome activation causing this punta formation, the NLRP3 punta formation. Just to illustrate, nigericin is a bacteriotoxin, is a control for a standard control for inflammasome activation used in the field. So you see a nice punta formation in response to nigericin. Then we measure cleavage of caspase-1 and IL-1 beta production, which was a readout for inflammasome activation as well. You can see here SARS-CoV-2 infection in primate cells triggers inflammasome activation. Nigericin is a control, so triggers caspase-1 activation, triggers IL-1 beta production, cleavage and activation of IL-1 beta. This can be inhibited by MCC 950, indicating that SARS-CoV-2 infection in primary human monocytes trigger LDH cleavage of caspase-1, cleavage of IL-1 beta, and punta NLRP3 punta formation, and also ASC punta formation. Just I'm not going to show here because of time, but also triggers ASC punta formation and NLRP3 punta formation. And also, I want to highlight this data because this is LDH. Remember, LDH was associated with severe disease in patients with COVID-19. So as a result of the parototic cell death, there is LDH release. So you see SARS-CoV-2 infection causes LDH release in the cells. But when we inhibited NLRP3 with MCC 950, we did not really decrease the release of LDH, which is suggestive that additional forms of cell death leads to LDH release independent of NLRP3 inflammasome. So to summarize this data, NLRP3 is activated by SARS-CoV-2. It requires live virus, active virus, leads to NLRP3 puncta formation, LDH release, caspase-1 cleavage, IL-1 cleavage, but also NLRP3 independent pathways for induction of cell death. This could be many different forms of cell death, necroptosis, other inflammasomes. So there's many other additional pathways that could be involved in this LDH release, which is independent of NLRP3 in this situation. So at that time, we decided to study and work with samples from patients from our university hospital, which is the HC FMRP USP. So we worked with 124 patients that were recruited from April 6th to July 2nd, 2020. So it was the peak of the first wave, peak of first wave pandemic in Brazil. In this period, we recruited 124 patients, which agreed to participate in the study. That's the characteristics and comorbidities of these patients, laboratorial findings, CRP, LDH. So all this information is going to be available, and it's published as supplementary data of this paper. So you can return there. But I just want to make the point that this case is where most of them moderate and severe COVID-19. What does it mean? So these patients were recruited because they seek for the hospitalization for healthcare. So this means these patients were more moderate and severe COVID-19. The patients that does not require hospitalization, they stay at home, they did not seek for care in the hospital, and they did not participate in this cohort of the study. Right, so this is more like the moderate and severe. 39% of the patients were moderate and 55% of severe. 27% of these patients had resulted in death because of COVID-19 during this study. So we recruited these 124 patients, and then we work with the samples from the serum of these patients. So, and then we measure by ELISA the cleavage of caspase-1. Remember, caspase-1 is produced as a proenzyme, is an inactive enzyme, but as soon as it's cleaved, there is an ELISA kit which specifically binds and senses the cleaved caspase-1. So we can measure active caspase-1 in the serum of these COVID-19 patients, and this is a log scale, so there is a huge production of active caspase-1. Also, huge production of active IL-18, so this is also cleaved IL-18, measured by ELISA, in COVID-19 patients as compared to controls, which is not COVID-19 or infections. So, and as you may know, IL-6 is a marker of disease severity, is a characteristic of COVID-19 as well. So higher levels of IL-6 were expected in this cohort of patients. And also we saw then a higher production of cleaved caspase-1 and cleaved IL-18 in these COVID-19 patients, suggestive of inflammasome activation in these COVID-19 patients. We decided to work with PBMCs from these patients. So we obtained a little sample of blood cells, and then we purified the PBMCs, excluded neutrophils, of course, and then we measure active caspase-1 by FLICA. FLICA is a dye that binds to active caspase-1. So this is from YVAD, and we can measure by FACS. So you see, this is a control patient. We see a very reduced FLICA binding in these PBMC cells, but when there is a COVID-19 patient, we see a super high activation of caspase-1, inflammatory caspase-1 or inflammatory caspases in this patient. This is just one individual, and this is a large group of patients. So in general, we see a higher activation of caspase-1 in PBMCs from COVID-19 patients as opposed to control patients. And we can see, if we just like get the PBMCs and put it on a slide and look in the fluorescence microscopy, stain it for anti-NLRP3, you can see the puncta, the NLRP3 puncta happens in many cells. This is not infection in vitro. This is patients from COVID-19 that we obtain a little sample of blood and then purify PBMCs, and then we can see active inflammasomes in the circulating cells of these patients. We also stained these cells with double-stranded RNA. During the replication of SARS-CoV-2, there's the formation of double-stranded RNA, and then we can see the cells are infected and there is virus replicating in the cells, and we see inflammasome activation, NLRP3 inflammasome activation in these PBMCs. We can also score the percentage of puncta formation. So this is anti-NLRP3 in PBMC cells. So you see many cells, there is a formation of this NLRP3 puncta. We also saw formation of ASC puncta. So, and in general, the COVID-19 patients has a higher percentage of NLRP3 puncta and ASC puncta as compared to the control individuals, which are not infected by SARS-CoV-2. And we also measured the caspase-1 activation by a luminescent kit. So there's a cleavage of the substrate by caspase-1. If there's caspase-1 present, means inflammasome activation. There's a cleavage of the substrate, and we can measure cleavage of the substrate by the cells of PBMC cells from COVID-19 patients. And also, we saw, although in this experiment was not statistically significant, in some patients we see a higher expression of IL-1, active IL-1, measured by ELISA. And then we decided to work with a group of pathologists, which are in my institution from the Department of Pathology and Legal Medicine. This is the Professor Alexandre Fabro, which is a fantastic pathologist. And he developed this method to make to generate minimally invasive lung autopsy, and he obtained samples from patients that deceased because of COVID-19. So there was only a small sample from the lungs of this patient were collected, and then we could perform like histopathological analysis and evaluate inflammasome activation and NLRP3 activation in these cases of fatal cases of COVID-19. So we see, first, this is a comparison of a patient that did not die because of COVID-19. So this is a control. You see the lungs is normal. And COVID-19 patients, you see a super inflamed lungs. There is a lot of cell recruitment to the alveoli and to the lungs of these patients. And we see we can stain these samples with anti-SARS-CoV-2, so we can see virus-infected cells in the samples. We can stain with the same regions with anti-CD14, so you see CD14 cells are infected with SARS-CoV-2, and they also express NLRP3, suggesting that the inflammasome is expressed in the NLRP3 gene or proteins, is expressed in the samples of lungs from COVID-19 patients. This is another example of a CD14 positive cell infected with SARS-CoV-2 expressing NLRP3. This so far does not mean activation of the NLRP3. This is just expression of NLRP3. But we then used two-photon microscopy of these samples to look for the activation of the NLRP3 puncta in this histopathological lung samples from COVID-19 patients. Then we can see this is a lung of a patient that deceased due to COVID-19. You see the nuclei stained in blue, and you can see this is the higher magnification of the cells. You see NLRP3 puncta activated in this lung of these patients. Another example, sometimes you see this NLRP3 puncta in the lungs of these patients. We can, of course, score the percentage of NLRP3 and ASC puncta in COVID-19 patients. You usually see a higher puncta formation, suggesting indicating activation of the NLRP3 ASC inflammasome in response to COVID-19. And also, we did a correlation matrix of all these patients, and we correlated many aspects and clinical characteristics of these patients, such as C-reactive protein, D-dimer, LDH, and also correlated this with the expression of active IL-18 and active caspase-1 in these patients. And as a proof of principle, we saw that in this group of patients, the higher activation of IL-18, of course, correlated to the higher activation of caspase-1, which makes sense. Caspase-1 is activated by the inflammasome, which in turn activates IL-18. So we saw a positive correlation of these two inflammasome markers. And also we saw positive correlation with IL-6 and caspase-1 and IL-6 and IL-18, suggesting that they are all producing the inflammation which is evident in COVID-19 patients. It's interesting to note that there is a positive correlation as well with C-reactive protein with caspase-1 and also with IL-18. And we also detected a positive correlation of LDH release with caspase-1 B20 cleavage, suggesting that they all are maybe associated with activation of the inflammasomes in these patients. This is a very busy slide, so don't try to read it, but this is just to show that we compared a lot of comorbidities and try to associate with inflammasome activation. You can see here that there is a correlation of IL-1 and 18, and we did not really saw any associations of inflammasome activation with the comorbidities of these patients. The only statistic significance difference that we detected was IL-18 activation in obese patients. So the patients which were positive for obesity showed higher expression activation of IL-18, suggesting that somehow obesity and inflammasome may be linked to activation of the inflammasome in these obese patients with COVID-19. This is a part of the, this is all this data is available in the manuscript. And then we finally move on to the last step of this investigation, that we try to associate mechanical ventilation with the activation of the inflammasome. And I have to make the point here that all these measurements of inflammasome activation were collected in the day of the internalization. The day the patients arrived in the hospital seeking for health attention, then there was collection of the serum sample. So all these points, each point represented an independent individual, the patient, and collected at the day of the internalization. So if the patient required mechanical ventilation, we saw that the inflammasome activation as measured by IL-18 in these cases were higher in patients that then required mechanical ventilation. Right? So the same thing happens with when we compare moderate and mild with severe COVID-19, and we saw that the patient with severe COVID-19 expressed a higher activation of caspase-1 B20, which is cleaved caspase-1, active inflammasome, as opposed to the patient with a more moderate and mild form of the disease. Unfortunately, we did not have the asymptomatic or patients that did not seek for health attention in this study, but it will be very interesting to see how is the inflammasome activation in these symptomatic patients with COVID-19. And finally, we divided this group of patients by the survivors versus the non-survivors, and the IL-18 was higher in the patients at the day of internalization. IL-18 levels were higher in patients that did not survive it due to COVID-19. So this is the 34 patients that did not survive it during this study, and then they has a higher inflammasome activation and caspase-1 activation. And this is a longitudinal analysis of some of these patients, and you can see the non-survivors, they usually arrive in the hospital with a higher levels of the inflammasome activation, as shown here by IL-18, and they reduce over the internalization in the hospital, but not as efficient as the patient with mild or critical and that recovered from the COVID-19. So, which was suggesting that inflammasome activation is participating on the outcome of the disease and somehow influences the outcome of the COVID-19 in this severe and moderate cases of COVID-19, suggesting that the inflammasome is worsening the cases of COVID-19. With that in mind, we start collaborating with a fantastic group of patients in group of clinicians in our medical facilities, mainly Paulo Lousada and René de Oliveira. They decided to pursue this moderate to severe randomized double-blind placebo-controlled clinical trial for colchicine. So colchicine is a drug very well used by rheumatologists to treat gout. Remember, gout is a disease caused by the NLRP3 activation because there is formation of uric acid crystals in gout patients, and these crystals, they damage the membranes and they cause a huge inflammation. And colchicine was in the acute crisis of gout, they provide to the patients colchicine, which inhibits tubulin and therefore inhibits the inflammasome activation, recruitment of neutrophils, and then the patients are treated by colchicine. So it's a very secure, safe, and not expensive drug for inflammatory diseases such as gout. And they established this clinical trial to test the effect of colchicine in severe and moderate cases of COVID-19 in the hospital. So this study was a very small group of patients. So they worked with 18 groups in the placebo group, 18 patients in the placebo group, and 17 patients in the colchicine group, and we monitored here is just illustrating the C-reactive protein over the time. So there is day 0, 2, 4, and 7 post-hospitalization, post-treatment, and the C-reactive protein, we remember, was associated with inflammasome activation as well, with the caspase 1 and IL-18. There was in the colchicine group, there was a reduction in the C-reactive protein as compared to the placebo group, and most important is the data for time of oxygen supplementation and hospitalization. This is the placebo group, this is the colchicine group, this is the requirement of oxygen, so mechanical ventilation for these patients, and there is an amazing reduction of the oxygenation time from 7 days to 3 days as compared to the placebo group to the colchicine group. So you see a huge reduction in the requirement of hospitalization with the treatment with colchicine, and we saw a reduction in the hospitalization time from 8.5 to 6 days in the hospital, which implies a huge reduction in the time of the hospitalization if the treatment were approved in general. So this was an ongoing study. We are now increasing the samples of this clinical trial. But this is one of the implications of the inflammasome activation during the COVID-19, the worst case of COVID-19. So that's what I had to tell today for you guys. That's the picture of Ribeirão Preto and the medical school. So you can see here the city of Ribeirão Preto. We are in the University of São Paulo. It's a state university in the São Paulo state, but not in the city of São Paulo. And then this is Ribeirão Preto city. You can see here the medical school of Ribeirão Preto. This is the hospital, and I'm very grateful for the fantastic team of researchers and the clinicians that got together to make this work possible. So that I have a lot of people to thank. All these people listed in this slide were author of this manuscript. Tamara was a fantastic graduate student. She was the first author of the paper, also with a lot of help from Kayla, Adrienne, Amanda, Samuel, Leticia, Augusto, Deborah, Warrison. And also they got help from the Center of Virology, which had all the P3 facility running, the School of Biology, Tiana and Fabio, School of Pharmacy, Fabiani, the pathologists, Professor Alexandre, Sabrina, Lee, Mayra, and the Center of Research in Inflammatory Disease, Fernando Cunha, Larissa, Thiago, and so on. And also the clinicians that were in contact with the patients to make this work possible. And this is the picture of the group. It's actually an old picture because now we cannot get together anymore, unfortunately. So no more lab meetings, no more retreats of the lab. but this is just showing this picture to show Tamara here. She's a fantastic, she was a first year graduate student, was joining the lab in the middle of the pandemic, and she did a fantastic work putting all these people to work together. And I'm very thankful for Tamara and for all the team for make this work possible. So with that, I would like to thank you guys for the attention. And again, Elena and Carla, thank you very much for the invitation. It was really a pleasure to be here in this fantastic seminar series that you guys put together with such a brilliantism in the last year. And I hope and I'm sure you're gonna have great talks during these years as well.
Thank you, Dario. Thank you so much for a very engaging talk and sharing your important discoveries on inflammasome activation and the very promising treatment. Thank you so, so much. Carla, indicate, the Twitter.
Yeah, thank you, Dario. That was wonderful. Thanks for sharing your efforts in this area. So before we close, we would like to remind our attendees that you can ask questions to Dr. Dario Zamboni through Twitter. So please go to the Twitter account of Global IMMUNOTALKS. You will find there a tweet that says, ask questions for Dr. Dario Zamboni here. Make sure you reply to that tweet with your question, and do not forget to mention the hashtag GlobalImmuno and @ZamboniLab, which is the account that Dario will use to answer the questions. So thanks again, Dario. It was a true pleasure to connect with you again and listen to all the new discoveries from your lab. And well, we look forward to continue being connected. Thank you.
Thank you so much, Dario, and see you everyone next week.
Yeah. See you everyone next week.